Question number 1. The first amino acid tRNA which initiate translation in eukaryote is methionine tRNA, formal methionine tRNA, tyrosyl tRNA, alanine tRNA. Correct answer is option A, methynyl tRNA. Question number 2. The first amino acid tRNA which initiate translation in prokaryotes is methionine tRNA, formal methionine tRNA, tyrosinal tRNA, alanine tRNA. Correct answer is option B, formal methionine tRNA. Question number 3. In eukaryotes, the 40S pre-initiation complex contains all the following initiation factor except elongation factor 1A, elongation factor 2, elongation factor 3, elongation factor 4. Correct answer is option D, ELF4. Question number 4. Eukaryotic initiation factor 4A, 4B and 4F bind to 40S ribosable subunit, 16S ribosable subunit, mRNA, amino acid tRNA. Correct answer is option C, mRNA. Question number 5. The codon which serve as a translation start signal is AUG, UAG, UGA, UAA. Correct answer is option A. AUG is the start codon in translation. Question 6. The first amino acid tRNA approach 40S ribosable subunit in association with ELF1A and GTP, ELF2 and GTP, ELF2C and GTP, ELF3 and GTP. Correct answer is option B, ELF2 and GTP. Question number 7. ELF1A and ELF3 are required for binding of amino acid tRNA to 40S ribosable subunit, for binding of mRNA to 40S ribosable subunit, for binding of 16S subunits to 40S subunit, to prevent binding of 16S subunit to 40S subunit. Correct answer is option D, to prevent binding of 60S subunit to 40S subunit. Question number 8. ELF4A process ATPase activity, GTPase activity, helicase activity, none of this. Correct answer is option A, ATPase activity. Question number 9. ELF4B binds to 3' chain initiation codon on mRNA, binds to 3' end of mRNA, binds to 5' end of mRNA, unbinds mRNA near its 5' end. Correct answer is option D. ELF4B is unwind mRNA near its 5' end. Question number 10. Peptidal transferase activity is present in 40S ribosable subunit, 16S ribosable subunit, ELF2 amino acid tRNA. Correct answer is option B, 16X ribosable subunit. Question number 11. After formation of a peptide point, mRNA is translocated along the ribosome by ELF1 and GTP, ELF2 and GTP, peptidal transferase and GTP, peptidal transferase and ATP. Correct answer is option B, ELF2 and GTP. Question number 12. Binding of formal methionine tRNA to 30S ribosable subunit of prokaryotes is inhibited by streptomycin, chloramphenicol, erythromycin, mitomycin. Correct answer is option A, streptomycin. 30. Tetracycline inhibit binding of amino acid tRNA to 30S ribosable subunit, 40S ribosable subunit, 50S ribosable subunit, 60S ribosable subunit. Correct answer is option A, 30S ribosable subunit. 14. Peptidal transferase activity of 50S ribosable subunit is inhibited by rifamycin, cyclohexamide, chloramphenicol, erythromycin. Correct answer is option C, chloramphenicol. 50. Erythromycin binds to 50S ribosable subunit and inhibits binding of amino acid tRNA, inhibits peptidal transferase activity, inhibits translocation, cause premature chain termination. Correct answer is option C, inhibit translocation. Question number 16. Puromycin cause premature chain termination in prokaryotes, eukaryotes, both A and B, and none of these. Correct answer is option C, both A and B. 18. 17. Diphtheria toxin inhibits prokaryotic elongation factor 1, prokaryotic elongation factor 2, eukaryotic elongation factor 1, eukaryotic elongation factor 2. Correct answer is option D, eukaryotic elongation factor 2. 18. The proteins destinated to be transported out of the cell have all of the following features except they possess a signal sequence. Ribosome synthesize them are bound to endoplasmic reticulum. After synthesis, they are delivered into Golgi apparatus. They are tagged with ubiquitin. Correct answer is this option D. They are tagged with ubiquitin. 19. SRP receptor involved in protein export are present on ribosome, endoplasmic reticulum, Golgi apparatus, cell membrane. Correct answer is option B, endoplasmic reticulum. 20. The signal sequence of proteins is cleaved off on the ribosome immediately after synthesis, in the endoplasmic reticulum during processing, in Golgi apparatus during passage through the cell membrane. Correct answer is option B, in the endoplasmic reticulum. 21. The half-life of a protein depends upon its signal sequence, N-terminus amino acid, C-terminus amino acid, prosthetic group. Correct answer is option B, N-terminus amino acid. 22. Besides structural genes that encode protein, DNA contains some regulatory sequence which are known as Operon, cistron, cis-acting elements, trans-acting elements. Correct answer is option C, cis-acting elements. 23. Inducer and repressor are enhancer and silencer element respectively, trans-acting factor, cis-acting factor, regulatory proteins. Correct answer is option D, regulatory proteins. 24. Cis-acting elements include steroid hormone, calcitriol, histone silencer. Correct answer is option B, calcitriol. 25. Silencer elements are transacting factor are present between promoter and the structural gene, decrease the expression of some structural gene and encode specific repressor protein. Correct answer is option D, encode specific repressor protein. 26. Transactin factor includes promoter, repressor, enhancer, silencer. Correct answer is option B, repressor. 27. Enhancer elements have all the following features except they increase gene expression through a promoter. Each enhancer activates a specific promoter. They may be located far away from the promoter. They may be upstream or downstream from the promoter. Correct answer is option B. Each enhancer activates specific promoter. 28. Amplification of dihydrofolate reductase gene may be brought about by high concentration of folic acid, deficiency of folic acid, low concentration of thymidine and amethotyrine. Correct answer is option D, amethotyrine. 29. Protein which interact with DNA and affect the rate of transcription process. The following structure motif: helix turn helix motif, zinc finger motif, leucine zipper motif, all of these. Correct answer is option D, all of these. Helix turn helix motif, zinc finger motif, leucine zipper motif. 30. Lake Operon is a cluster of genes present in human beings, E. coli, lambda patch, all of this. Correct answer is option B. Lake Operon is present in E. coli.